All right. Well, good afternoon, and my name is Feline Bazin, and I'm a board member of Access, and I'm happy to welcome you all here this afternoon. I just want to introduce our next speaker, Dr. Armin Raznahan. And Dr. Raznahan is a child psychiatrist who leads the NIMH National Institute of Mental Health study on X and Y variations. The National Institute of Mental Health group aims to better understand X and Y variation efforts on the developing brain to help answer families' questions about development and well-being. So this session is updates from the National Institute of Mental Health Intramural Research Program study on XY chromosome variations. So welcome, Dr. Raznahan. Thank you.
Hi, everyone. Thank you for coming to the session. It's great to be here with you all. And thank you for that introduction. So as you were hearing, my name is Armin Raznahan. I'm a child and adolescent psychiatrist, and I lead the NIMH study on XY variations just outside Washington, D.C. And I wanted to take a few moments of this session just to tell you a little bit about our goals, what the study looks like, and some of our recent research outputs and plans for the near future. But before I dive in, just wanted to give two big shout outs. The first is to the individuals with XY variation, their family members, the community here. The work that I'm about to present would have been absolutely impossible without the incredible support and involvement of the community here. So it's really, we're so grateful to you all for being part of this. And also a huge shout out to the community of clinicians and researchers. As we were hearing this morning, what a dazzling display of the diversity and the vigor of the work that's happening in the space. And as Nicole was saying, as Dr. Rantaglia was saying, to see it grow like this has just been incredible, even in the short time that I've been involved with it. So what I wanted to do today was just to tell you a little bit about what the current study, how it came about, who's on the study team, what are our main goals, what's the study design, and what is it like to be a participant in the study. I'm going to give you a selection of just some of our recent findings, just to give you a flavor of what we do, and then let you know who to contact for more information. So an overview of how the study came about. So this is the NIH Clinical Center in Bethesda, Maryland, just outside Washington, D.C. And the first phase of research on XY variations at the NIH was started by Dr. Jay Geed, and it ran between 1990 and 2010. And that study mainly focused on cognition and brain anatomy. And it looked at about 150 participants who were aged between 5 and 25 and represented a whole variety of different sex chromosome aneuploidy carrier types, combinations of X and Y chromosomes. This new phase of the study that we're currently in started in 2015 and sought to build on the incredible work that Jay Geed had started. And what we wanted to do is we wanted to expand the size of the study, so to be 3 times larger with about 500 participants per time point, and to really get much more detail in the measurement of cognition, behavior, and development that we gathered. And the richness we wanted to get as we were assessing how the young people in the study were doing is both so we could give the families more detailed feedback reports, but also so we had more granular information for the research projects. And unlike the first phase of the study, we also gather information from the family members. So we assess behavior and cognition in the parents who come in with the young people in the study. And we gather more detailed measures of the brain and the genome in this phase than in the initial phase. So who's on the study team? We're part of the study currently and have been part of it in the past. Some of you in the room may work with some of the research workers who've come through the study and gone on to become psychologists, doctors, and scientists. So Dr. Torres is the nurse practitioner within our study and does many of the mental health assessments. Shrishti Rao and Jessica Seebeck, Jessica's with us here, are the amazing clinical psychologists on our team who help plan the cognitive assessments and do the detailed assessments of autism features, autism-related features. Leave Clarkson is our database manager. Francois Lalonde helps gather the brain scans. And we have Shara Raymer and Sienna Molestuen who are with us here, who are the current generation of research workers within the project. So they'll be interacting with the families on the sort of front line. And then here's the sort of aged old photo of me. And as I say, behind the scenes, we have collaborators in the areas of genetic counseling, of reproductive endocrinology, and a whole team of scientists who help work on the rich data that we are lucky enough to be able to gather to understand how X and Y chromosomes can impact human development. So our goal is to advance clinical care, is to use data to advance clinical care. And the way we structure our goals really is informed by the questions that families come into the protocol with. So we often hear, as we've heard already today, a common experience of individuals with XY variation is we have to tell the doctor what this XY variation is, what XXY is, for example. And to try and address this need, our goal is to increase awareness of XY variations amongst clinicians and scientists, and to improve our understanding of development in XY variations and to share this with the community. So what we do here is we take all the cognitive and behavioral data and we write reports that really detail as finely as we can what sorts of issues people with each type of XY variation can experience. And then we publish those with the idea of disseminating them so that hopefully if someone reads that, then when they meet a new family that's been diagnosed, they'll have more accurate information to share with them. A second set of questions we often hear is, what can we do to help support our child's development? Should we consider treatment A or treatment B? And what essentially this has to do with is variation in outcome. Within any one XY variation group, say XYY or XXY, everyone is an individual. No one is affected exactly the same way. So you can have a huge diversity of outcomes within each individual XY variation group. So what we try to do is to understand that variation. How large is that variation? What factors can predict which people seem to do better and which people may have more challenging problems? So a lot of our work is trying to identify predictors of outcome, and I'll give you some examples of that later. And then finally, they're the kind of why questions, the mechanistic questions. Why is our child having these developmental difficulties? Are there biological tests we can do early in their development to sort of predict where they might end up? And this is really, I think, about understanding how X and Y chromosomes influence human brain organization and how they do that by influencing the genome. Ultimately, the process has to be that when a cell is carrying extra X and Y chromosomes, the impact of those chromosomes on the genome is the first step that then can percolate through development to give some people some challenging outcomes. But as I said, there's a wide variation. So having an XY variation doesn't dictate with certainty that a person is going to have difficulties. So what's it like to be in our study? What's a study design like, and what's it like to participate? So our study design is shaped by 4 guiding ideas. The first is that we can do a better job of understanding any individual XY variation, say XXY, by studying multiple different XY variations. So there are really important reasons for doing focused studies on individual groups. What are the needs of young people with trisomy X? What are the needs of young people with Klinefelter syndrome? And that work is important. But there's also real value in combining information from those 2 groups, because both individuals with trisomy X and individuals with Klinefelters carry an extra X chromosome. So by studying 2 different examples of carrying an extra X, we can get a clearer picture of what an X chromosome might be doing to development. A second guiding principle is we have to understand individuals as well as groups. And this is something I was saying earlier. Variability is the rule. There is no one-size-fits-all picture of a person with Klinefelters, for example. So some of our studies try to describe the average outcome in a group of people that have a particular XY variation. But then we also need to do work to understand the variability in the outcome. And I think a third guiding principle in the study is we need to get better at measuring development. So if you think about how people in the field of mental health measured development, back in the day, it was very much in these diagnostic bins. You know, do you have diagnostic boxes? Do you have autism? Yes or no? Do you have ADHD? Yes or no? But I think we need to go beyond that. because we all really exist on a spectrum. There's not something magical that happens when you're one side of the diagnostic box or the other. So I think it's really important, we as a group feel it's important to use very fine-grained measures of behavior rather than just yes-no measures, and to measure multiple aspects of behavior at the same time. Because for each of us, the way we experience the world is like a fingerprint. So you need to measure all of those ridges to really get a complete picture of how a person is doing. And then I think a fourth guiding principle in the study is it's really important to study behavior and cognition, because these things can most immediately impact a person's day-to-day life. What we also want to do, though, is be measuring the brain and the genome to try and understand how does a given person arrive at a particular profile of strengths and difficulties. So to do that, we measure data at multiple levels. So we have to measure how XY variations influence genes, how they influence the brain, and how they influence behavior. So I'm giving you some examples of this. This is a gene network. Each of these dots is a gene. And these genes all share the feature that they change their expression when you carry an extra X chromosome. So we're building this module of genes that all move together when you have extra X dosage. And these are the kind of candidate genes that could be driving the effect of the X chromosome. This is an example of some of our neuroimaging work. This is a spinning brain, the outside of a brain. And where you see a color, these are the cortical regions that on average tend to vary in their anatomy when individuals carry extra X or Y chromosomes. The red regions on average change their anatomy with an extra X or an extra Y. The green regions are uniquely sensitive to X dosage, and the blue regions are uniquely sensitive to Y dosage. So if you think about it, I was describing at the level of behavior how it's important to study multiple XY variations together, because we want to figure out what challenges and strengths are shared across all different XY variations and which are unique. And this is that same question, but now in the space of the brain. Which brain regions are always affected, whether you have an extra X or Y, which might be specific to different syndromes. And this is an example from a recent study of behavior where we compare different XY groups for how different behaviors hang together. I'll come back to that in a moment. So to get these data involves a long visit. So this is what a schedule might look like for a person participating in the study. This is for an individual with XXY syndrome. Sorry if the slide's a bit busy, but this is Monday and this is Tuesday. So families will come in, they'll be welcomed to the NIH Clinical Center, they'll be admitted, they'll be consented for the protocol and start doing some questionnaires. Then they'll get their blood pressure and heart rate and weight, height and weight measured. And then there might be an assessment of autism features and neuropsychological profiles whilst the young person is practicing the MRI scanner, for example, and then there can be tests for endocrine development, a DEXA scan to assess bone age, interviews with the parents to get a sense of what their experience of the young person's development is, consult with endocrinology, and then the MRI scan towards the end. So you can see it's quite a busy couple of days, but the information we gather is used for two main reasons. The first is this multidisciplinary team review. So every 2 weeks, all the individuals who work on the protocol get together around a table, and we spend time talking about each individual who's come through the study, talking about the various assessments, so that we can share knowledge and come up with a final set of recommendations for the family in a written report that we share with the families that come through, that detail the results of the test, give a summary where we describe the areas of strength, the areas of difficulty for that young person, and then end with some recommendations as to the extra care that that person should be seeking when they go back to their home state. The second output from those 2 days is that all the data that are gathered go into the research analyses. And to give you a flavor of that, I just wanted to give you some example of recent findings from our study that use the data that have been gathered in the second phase of the study. So the first sort of question we can ask is, what aspects of mental health are most impacted in each XY variation? And this example I'm giving is from XYY syndrome. So these are results from 64 individuals with XYY syndrome. And what we wanted to understand is we wanted to measure a whole bunch of different aspects of behavior. And for each aspect of behavior, measure the average score on that aspect of behavior and the variability. So let me walk you through this plot because it's a bit of a complicated one. Each of these bars relates to a different measure of behavior. And the name at the bottom is an abbreviation of that measure. So for example, this SOC CBCL, this measure is the social difficulties scale within an instrument called the CBCL. This measure, SOC SCQ, is the social problems score from another instrument, the SCQ. So this is about 60 plus different measures. And I've color-coded these measures by the area they have to do with. So all the blue ones measure sort of social difficulties. The pink ones are like total scores. If you measure all the mental health difficulties and just put one number, that's what the pink scales estimate. The tan ones measure externalizing difficulties. This is sort of rule breaking, irritability, aggression, these sorts of things. The green ones measure internalizing difficulties. This is sadness, anxiety. The red ones have to do with cognition, and these light blue ones are repetitive behaviors, being fixed on certain patterns. And what I've done is, so what you see in this distribution for each of these bars, this is the range of scores that we saw in the 64 individuals with an extra Y chromosome. The very top of the line is the highest score, almost the highest score we saw, the 95th centile. The very bottom of the line is the lowest score we saw, the 5th centile. And then this bar covers the 25th to the 75th centile. And you see these lines, this is the mid number. You can think of it as sort of an average score in that aspect of behavior. And the higher up you are on this scale, the more severe the difficulty. So this individual up here, this individual who's this dot, has really a very high score for social difficulties, whereas the people down here at the bottom have very low scores. They have low levels of social impairment. This line, 0, this red line, is the population average. And this dotted red line is what's considered the threshold for a high score in the average population. And what I've done is I've ranked all of these measures by how severely impacted they are. So what this lets us do is systematically, if we measure 60 different behaviors and then we order them, it gives us a really clear picture of, on average, in individuals with XYY syndrome, what aspects of behavior tend to be most impacted and which ones tend to be least impacted. And knowing this is really important for individuals who have XYY, for their family members, and for the healthcare providers who work with these families. Because if you know that in individuals with XYY, certain things are more impacted than others, that can help you target your conversation with that young person, helps you tailor your assessment. So what this shows is that we find in XYY syndrome, some of the aspects of behavior that tend to be most severely impacted have to do with social functioning and cognition. Whereas some of the areas that are least impacted are these externalizing scores. So these bottom ones, these are hardly impacted at all. And these are measures of aggression and hostility. And that's really, really important because one of the major issues with XYY syndrome is this awful incorrect stigma that's associated with the condition that has to do with flawed research in the past that led people to think that having an extra Y chromosome was associated with aggression and a tendency to violent behavior. And this study very clearly shows that's actually the least of the issues. That is one of the areas that isn't really impacted. So this is an example of one type of study where we're mapping the profile of strengths and difficulties in each aneuploidy group. A related question you can ask is, this plot I've just shown you, this is for XYY. You might be sitting in the audience saying, well, what does it look like for XXY? Is it the same? Is it different? Are there some behaviors that are more impacted when you have an extra X and some that are more impacted when you have an extra Y? Well, what we do to answer that question is we do exactly the same thing that I've just shown you here for individuals with XYY. We do the same thing in individuals with XXY, and then we compare. And that's what I'm showing you in this plot. Let me walk you through this plot. It's a little bit complicated. Each of these dots is one of those measures of behavior. And I've colored them again by the domain that they measure. So all the blue dots have to do with social difficulties. The green dots have to do with mood and anxiety difficulties, for example. The x-axis is how severely impacted that behavior is when you have an extra Y chromosome. The further to the right you are, the more severe on average are people's difficulties with that measure. And the y-axis is how severely impacted that measure of behavior is when you have an extra X chromosome. So the way to read this plot is like this. The further to the right you are on this axis, or the further to the top you are on this axis, the relatively more severe the impact of the aneuploidy on that aspect of behavior. So what this tells you is if you look at XXY, the most impacted scales, the ones highest up on the y-axis, have to do with overall mental health symptoms, but anxiety, depression, and internalizing mood difficulties. If you look at XYY, the ones that are furthest to the right, these blue ones have to do with social difficulties. And that tells you that relative to all the other measures, social problems tends to be a particular area of sensitivity in XYY syndrome. And with this type of correlation plot, if XXY and XYY had exactly the same impact on behavior, all of these dots that you're seeing, can you see this little dashed line here on the diagonal? If these two syndromes had exactly the same effect, all the dots would be lined up on the diagonal. But they're not lined up on the diagonal, right? The first thing you see is they're scattered. And then the second thing you see is they're actually all a bit below the diagonal. And this gives us two important pieces of information. This tells us that on average, XYY seems to have a larger impact on mental health symptoms than XXY. For any given aspect of mental health measurement, the score of difficulties tends to be a bit higher in XYY than XXY. And it also tells you that, but there's an exception, which is these mood features, which are equally impacted by an X and a Y chromosome. So this tells you that social problems are the most differently impacted by the two syndromes. They're particularly more impacted by Y, whereas unusually mood symptoms are impacted just as much by an extra X and an extra Y. And we think this type of, and whereas all these dots down here, they're not impacted that severely at all. So we think this is really important because what the question this addresses is, if you're a doctor and a family come in, and you know that one child you see is carrying extra X, whereas another child you see is carrying extra Y, this sort of data tell you, should I be digging deeper into particular areas for those two children? Should I expect the same outcomes, or are there particular areas I need to go into with more detail when I'm seeing an individual with an extra X or an extra Y? Sorry. So this is about precision medicine. This is about personalizing the questions you ask. So takeaways from this: not all aspects of mental health are equally impacted by each XY variation. On average, social and learning skills tend to be most impacted. These impacts tend to generally be a bit more severe with an extra Y than an X, but that doesn't apply to mood features, which are equally impacted. And we're now doing exactly the same thing for trisomy X syndrome. But a really important point to drive home is these little dots you see are just the average. Do you remember that last slide I showed you? This big spread. That's really important to hold in mind, that every individual is unique. And just because you have a particular XY variation, it doesn't mean you're definitely going to have a difficulty in a particular area. Everyone has their own profile of strengths and weaknesses. Each child is unique. And there's also lots of room for change. This is just one snapshot in time, whereas we all change as we grow. So it's a dynamic system. We're just describing it at one moment. So then the challenge becomes understanding that variability. Can we do any better than we're currently able to do at predicting how impacted a specific child with an XY variation might be? And a couple of years ago, we published a study addressing this question with specific reference to this was an XYY syndrome. And this particular plot that I'm showing you as an example has to do with language outcomes. So let me walk you through this plot. Each dot is a child. And the x-axis is the family score, the average score of the family members of that child in terms of their language functioning. And the y-axis is the score of the child on the same test of language functioning. And if children and their families had precisely the same language score, all of these dots would be lined up on this diagonal. But what we see is they tend to be shifted down from the diagonal a bit. And there's a correlation between the two scores. So this tells us two bits of information. The first thing it tells us is, compared to their own family members, you tend to have a slightly lower language score. The individual with the XY variation pretty consistently tends to have a lower language functioning than their family members that don't have the XY variation. But it also tells you that there's loads of variability amongst people with an extra Y chromosome in language. So the average score is 10 in the population. So we've got people in our study who have higher than average. They have an extra Y chromosome, but they have higher than average language functioning. And there are some people in our study who have lower than average language function, but there's a big old range. And a newborn individual, as we were hearing, the growing detection of individuals prenatally, with a newborn individual in that range later. So we want to try and predict that. And the fact that there's this diagonal line here tells you that you can kind of predict the outcome in a child by measuring the score in their family member. So the idea here is that when a young person is born, if you're able to measure the language functioning in their parents, that doesn't dictate what the young person's language functioning is going to be, but it helps you get a little bit better at guessing where that young person is going to end up. And we think that's really important because for now, if you were to be newly diagnosed and go to someone and say, my child has an extra Y chromosome, how might that impact their language? The best guess that the field has right now is the average language score in individuals with an extra Y chromosome. And what this lets us do is do a bit better than that average, try and do a child-specific prediction. So this was an early study in XYY looking at language. We repeated this study recently in a larger sample of XX individuals with XXY syndrome. And Shara Raymer has a poster on this. So if you want to learn more, you can go and chat with her in the poster room. This is the same plot as I've just described, except this is in XY syndrome. And this measure is general cognitive ability rather than language. This is sort of also referred to as IQ. And again, we see that there's this positive correlation. So on average, the group of individuals with XXY syndrome had an IQ of 90 on average. But you can see a massive variation. Some people in our study have an IQ of close to 130. That's extremely high general IQ score relative to the average. But some people have lower scores. So this is, again, an example of how you can use family data to predict child outcomes. So it seems that so far, comparing the two syndromes, that cognition is the one that we tend to be able to consistently predict best. So we think general cognitive ability may be an outcome in individuals with XY variation that we might one day be able to predict from family scores. But I want to be really clear that general cognitive ability is just one aspect of development. It's only one aspect. It's got some importance, but we aren't our IQ scores. We're a hell of a lot more than our IQ scores. But I'm using IQ as an example of one outcome that we can measure. Where an individual child is going to be, but there's still error. You can't perfectly predict where a child is from their family scores. Therefore, we need to do more work to apply these sorts of measures in real life. And the other important thing is it's just a prediction. Prediction is not set in stone. This is a mathematical model that lets you make a better guess at where a young person might end up, but that's not prescribing a fate to that young person. There's a whole lot of living and learning and experience that can change the developmental course that someone follows. Nevertheless, though, we think it's important to be able to do this sort of prediction, because if you have a young person who, based on their family scores, for example, is predicted to have more severe difficulties, then that could be a young person you want to sort of watch more carefully as they develop and give extra input to. Okay, so I've described what's happening, some of our recent work on cognition and behavior. But another set of questions that we ask, as I was mentioning, has to do with the biological mechanisms that might be underpinning these impacts of XY variations on cognition and behavior. And to do that, we have to measure the brain. And we do that using MRI. So we take MRI scans. The whole scanning session can take about an hour. And then we take those pictures of an individual's brain and we process them through a computer to measure different aspects of their brain organization. And I'll walk you through this plot. It's a little bit of a complicated XY syndrome, and below the line has to do with XYY syndrome. On the top slide, at the top brain is the left-hand side of the brain from the outside, and this lower one is the inside of the left-hand side of the brain. So each column is a different measure of the brain. So this first column has to do with how thick your cerebral cortex is. So if you look at someone's brain, it looks like a walnut. And the outside of that walnut is a thin layer of cells, about 2 to 4 millimeters thick. And that's the cerebral cortex. And what this map is showing you is the differences in the thickness of that sheet between individuals with XXY and XY individuals. So boys and young men without an extra X chromosome and boys and young men with an extra X chromosome. So where you see red, sorry that these brains are so small, but where you see red, the cortex tends to be on average thicker when you have an extra X chromosome than when you don't. And where you see blue, the cortex tends to be on average thinner when you have an extra X chromosome. So I've just walked you through what these two plots are telling you. These are changes in average cortical thickness when you have an extra X. This is the same thing when you have an extra Y. So if you compare these two, they look different, don't they? Right? You've got a whole load of blue here that's very dark and less blue here, for example, or there's a red dot here and it's not so red here. But at the same time, there are some similarities, right? The front and the back tend to be redder, the front and the back tend to be redder. So what this is telling you is that just like I was saying with behavior and cognition, there are subtle differences between the impact of an X and a Y chromosome on behavior and cognition. This is telling us that there are subtle differences in the impact of an extra X and Y chromosome on one measure of your brain, the thickness of your cortical sheet. But our brains are super complex, right? There are loads of things you can measure about our brains, not just how thick the cortical sheet is. You might want to know how bendy is the cortex, how curved is it? That's what this map tells you. How much volume is there? What's the area? So each of these columns is a different measure of the brain. These ones here, for example, have to do with white matter, the wiring that connects different brain regions, how the white matter properties of the brain changed. So you can think of this map. These are 15 different columns. These are 15 different measures. So it's like a fingerprint on the brain, essentially. We're trying to say, what is the brain change fingerprint on average of carrying an extra X? And we measure 15 different properties of the brain. There are 15 different curves on the fingerprint, if you like. And then we do the same thing for having an extra Y. What is the average fingerprint of having an extra Y on the brain? And there are 15 different curves. So when you look at this from a distance, a couple of things pop out. One is that these maps vary as you move along the column, right? So different brain features are impacted in different regions in different ways. It's not just everything changing the same way at the front, everything changing the same way at the back of the brain. And then the other takeaway is these columns don't look the same. You see, this is the impact on curvature with an extra X, but there's a very different impact with an extra Y. So this is suggesting that the X and Y have some shared effects on the brain and some different ones. And this is a lot of information, right? These 15 maps. And we use a method called principal component analysis you can think of it as averaging, if you like. We take all of these 15 different measures and we average it into one grand summary map. And this is the grand summary map for XXY, and this is the grand summary map for XYY. And the way to read these maps is like this. Where you see a deep color, where you see a deep red or a deep blue, that's a part of the brain that seems to be changing quite prominently because of the extra chromosome. But where you see white, these are brain regions that really are unaffected. They actually kind of look, on average, exactly the same. It doesn't matter whether you have an extra X or an extra Y. So you can think of these maps as kind of road maps of the brain's sensitivity to sex chromosomes. And we think these road maps are important, and we're making these road maps for each aneuploidy syndrome, each subtype, because we think that if we can figure out which regions are red and which regions are blue, that tells us the systems that might be relevant for the kind of behavioral and cognitive issues that I was just presenting. So what we want to understand is by mapping the brain, it first tells us which brain systems could be contributing to the day-to-day difficulties. That's one clue it gives us. But the other clue is it tells us where to look deeper. Because if in a given brain, one region seems to be really sensitive to whether you have an X, and one region really couldn't care less, it's not changed at all, the question becomes, well, what is different about those two regions? If we can figure out what's making the blue region vulnerable while the white region doesn't care, that could be a clue about how X chromosomes influence development. And one of the big mechanisms we're interested there has to do with genes. How much time have I gone? Okay, I'll wrap up so this time, but we've got 10 more minutes, right? Okay, I'll wrap up so we have time for questions. And then this final slide is doing the same thing, but dropping down to the level of genes. So we have about 20,000 genes, each of us. So you can say the same thing. In the brain, you might say this brain region is more sensitive to an X chromosome, this brain region isn't. In the genome, of the 20,000 genes, you might say this gene really changes its expression when you have an extra sex chromosome, but this one actually doesn't change at all. So that's what we're trying to figure out. So this is a plot from a study that's under review right now where we gathered data that other people had published over the past 20 years or so, which encompasses about 400 individuals with sex chromosome variations. And each of these big dots, these are genes that really change their expression when you have an extra sex chromosome. And these little lines is telling you that these genes work together. It turns out the genes that sex chromosome aneuploidies impact actually sit within networks that operate together. They're not just random genes. They're genes that tend to be involved in some similar things. So we're zeroing in on the genes out of the 20,000 that seem to be most sensitive using old published work. The second thing we're doing is studying gene expression in tissue from people with XY variation. So the amazing people who've come through our study have given skin and they've given blood. We've changed that skin and blood into stem cells, like unprogrammed cells that can then be made into any other type of cell. And we've taken these stem cells and we started to turn them into different types of brain cell. And now we're able to measure XY chromosome influences on gene expression in models of the human brain. It's very difficult to get to the living human brain because we all need our brains and they're protected in our skulls. So this is a way of trying to get around that by taking skin and making kind of brains in a dish and trying to understand gene expression that way. So just to wrap up, just to give you a sense of the work underway, everything I've just told you is largely focused on XYY and XXY syndrome. We're now doing this work with the incredible community of trisomy X, individuals with trisomy X who've come through our research study. We're going to continue comparing the different syndromes, and we're going to be focusing on how genes and hormones can influence variable outcomes. And we're super excited to be part of some of the other incredible studies you've heard about, Galaxy, and hopefully soon to be funded NASCAR, and other European collaborations. Because if, one of the big messages from today is the way we make maximum impact is by joining forces. So we're really thrilled to be part of that wider community. And if you want more information, please contact Dr. Erin Torres, who's a nurse practitioner, or Rachel Gore, who is the clinical genetics colleague within our team. And the most important slide of all, before I take questions, is this one, which thanks you all again and Access for making this work possible. Will be completely impossible without you all, and the amazing team of colleagues and scientists and trainees who've helped actually do this work. Thank you very much.